Okay, good afternoon, everyone. Welcome to the Biophysics and Mechanical Biology seminar. So today it's my great pleasure to talk about our work on mechanics and geometry. So I actually added the tunable helical ribbons part because I think it gives you a flavor of how I started this journey on mechanics of shape formation and evolution. And then I'll be talking about embryonic brains, torsion, biohybrid soft robots, and collective cell migration. So just a very brief introduction of solid mechanics. This can be dated back to Galileo, who studied the beam problem. And Robert Hooke, proposed this famous Hooke's law. So basically F equals K delta X, right? So force is linear proportion to displacement. And there are other great scientists like Bernoulli, all studied mechanics problems. And to appreciate this talk, all you need is love. So the first two L refers to Landau Lifshitz, who wrote this very nice little book, Theory of Elasticity. And for those of you who are math savvy, you're welcome to check out the book by Love. But really what you need to know is just a little bit of a linear elasticity theory. So stresses are linearly proportional to strains. So this is basically a 3D version of the Hooke's law. So the stress in this case is written in a form of matrix, and so there's a strain. And so the strain here is defined as the following partial derivatives. Oops. Shown here. And in linear elasticity theory, we basically ignore the higher order terms. So this is the approximation. So here U is the displacement vector. So if you started, for example, with a rounded shape, right? So a position here, a point here, under deformation, under mechanical stress, it deforms into a shape of heart and it moves to another point. So by subtracting these two vectors, you get the displacement vector. And then by taking partial derivatives, you can arrive at the strain tensor that quantifies how much this body is deformed in 3D. And then constituted relationship simply states that stresses are linearly proportional to strains. They're related by this elasticity tensor. And so I started by studying the formation of helical structures because they're almost everywhere in nature. And it's an interesting shape because it breaks the left-right symmetry. They're everywhere in nature, going from that DNA double helix, alpha helix, they by layers, bacteria flagella, these microscopic objects, to macroscopic things like horns of animals or tendrils in plants, and seed pods. So these seed pods actually were found outside of Dartmouth, where I worked previously. And engineers have also been very interested in studying nanohelices that can be used as resonators, sensors, transducers, transistors, and biomedical devices. And the problem that I started out to study is how do we get a helical ribbon shape out of an initially flat straight ribbon? Well, you can say it's very easy because we can simply bound it to the surface of a cylinder. So in this case, the cylinder lies horizontally, and it has a radius 1 over kappa 1. So kappa is one of the principal curvatures. Or alternatively, you can bound it on the surface of a cylinder that lies vertically with presumably a different curvature. An interesting question is what happens if this ribbon wants to bend both along, the horizontal and vertical axis, and presumably with different principal curvature. So would it become left-handed or right-handed, or some, one or the other? So in this case, we found that it will, take a helical shape form, but in doing so, the edge of the ribbon is no longer straight, just like this lipid bilayer it's either concave or convex. So in fact, a one ribbon, if you zoom in, it forms it locally into a cap shape with principal curvature kappa 1, kappa 2. And the principal bending direction can form an angle with the geometric axis dx and dy along the length and width direction of the ribbon. So there are these three independent geometric parameters, kappa 1, kappa 2, and phi, that controls the shape spatial configuration. If we're given these three, mathematically, we can find out the 3D configuration of the ribbon written in terms of kappa 1, kappa 2, and phi. So they're related here, and S is the arc length that's measured from the beginning, from the origin of the coordinate system. Now, if we plot them, I'm showing you here, just by changing the relative values of kappa 1 and kappa 2, and phi is fixed to be 45 degrees, then we can get a range of geometric objects, such as rings when kappa 1 equals kappa 2, or a cylindrical helical ribbon when kappa 1 or kappa 2 is 0. But in general, they form a helical ribbon shape when kappa 1 does not equal kappa 2. And when kappa 1 is equal to negative kappa 2, it forms a purely twisted ribbon. So just by tuning these geometric parameters, you can get all these shapes. The next question is, how do we get these shapes from the mechanical parameters, such as Young's modulus and Poisson's ratio? For that, I built a mechanics model. Without going into the details, we were able to write down the total energy associated with this deformation. And that takes into account the in-plane strain and the strain due to bending and the residual strains. And then we can minimize the energy to find out the unknown parameters and get analytical solutions. And we can show that the set of solutions, when the Poisson's ratio becomes 0, it can be reduced to the classical Stoney's formula. And of course, if it's only theory, is not so interesting or convincing, and that's why we also did experiments. And so we did these tabletop experiments to fabricate these ribbons using a string engineering approach. So the way we did it is we pre-stretch a rubber sheet in perpendicular directions. So we can tune up the pre-stretch in 2 directions, and then we bond an elastic ribbon along a tunable angle. So the 3 parameters we can tune, they're bounded together and let go, and it forms spontaneously into a helical ribbon. And I'm showing you the theory versus experiments when the driving force is anisotropic. So the pre-stretch in the perpendicular in the vertical direction is twice as the in the horizontal direction. We got pretty good agreement. And we can, so basically when the misorientation angle phi is 0 or 90 degrees, we get ring shapes. But otherwise, the helix pitch and the radius would depend on the 3 parameters. And then we also show that if the driving force is isotropic, then we get ring shape regardless of the misorientation angle. And this is again consistent with the theory, because if we calculate the helix angle, when kappa 1 equals kappa 2, because the driving force is isotropic, then regardless of the misorientation angle, this is always 0. And arctangent 0 is 0. That means it's a ring shape. And we can also obtain a purely twisted ribbon shape, and that's through a 3-layer experiments. So we pre-stretch the top and bottom layer, and then sandwich a elastic strip in between at 45 degrees. And according to the theory, in this case, kappa 1 equals negative kappa 2, because locally it prefers a saddle shape. But globally, it takes on a purely twisted shape, because when phi is 45 degrees, this is 1/2, and that is also 1/2. And if you calculate r, the radius of the bounding of the helix, it becomes 0. So it's twisting around its axis. And so interestingly, if we keep exactly the same pre-stretch for this ribbon, but we just change the dimension, so if we make it wide enough, then it changes shape. So it changes from a purely twisted ribbon to a nearly cylindrical helical ribbon with a 45 degrees helix angle. And more interestingly, now it becomes bistable, so it can take on a second stable configuration. And similarly, for this ribbon with the 30 degrees misorientation angle, when we increase the width, when it goes beyond the threshold value, it also becomes bistable. So previously it was monostable, it only has one stable shape, but now it can be stable in this configuration or in that configuration, also shown in this purple rectangle. And so this is a video showing the transition from one helical ribbon shape to the other. So it's a bistable configuration, and it can change shape in response to external stimuli back and forth. And so the same thing, and we can apply the same mechanism to a stainless steel bar. So this is, you can see, it's a measuring tape. So we can make it so that it has two stable configuration. It can switch between these two stable configurations in response to external stimuli. So now if I'm presenting this with you in person, that I would have brought these samples with you. And but if you play with them, you have to be very careful, because this bistable strip, it changed shape so fast. Sometimes it cut my finger, and that's why I was joking with my students that this is direct proof that we're working on cutting edge research. Okay, so this type of transition from a purely twisted ribbon to cylindrical ribbon, cylindrical helical ribbon, actually occurs in a wide variety of physical systems that include liquid crystal elastomers when the temperature changes, or in Alzheimer-related peptides when the dimensions change. So it goes from a purely twisted ribbon to a cylindrical helical ribbon, and then to a tubular structure. And that shows the agreement between the theoretical prediction and the dots are from the experiments from our collaborator at Emory and Georgia Tech. Just quickly sum up, we developed a theory that interprets the spontaneous formation of helical ribbon shapes due to mechanical anisotropy and geometric misorientation. And we showed that the smooth transition between these geometries can be tuned and controlled via external stimuli, and that can imply applications in sensors, transducers, drug delivery systems, or robotics. And to move on to the biomechanics of shape formation in embryos. So in the past century, the scientific community has celebrated the huge success of the molecular genetics studies of development, but still a lot remains to be understood about how the one-dimensional information coding in DNA can be translated through mechanics into three-dimensional biological shapes, a process called morphogenesis. So these embryos early on, they look pretty similar in shapes, but later they can develop into dramatically different organisms. And there have been very elegant studies on the roles of mechanical forces in the formation of guts, for example, Cliff Tip and Hartman. So they studied also using a rubber model to replicate the looping guts that's observed in avian embryos. That's due to differential growth and the mismatch between the tissues. And so here we've been trying to understand the formation of the early brain shapes in hope that our study can inspire further understanding of congenital brain defect formation that occurs in approximately 1% of live human births. And it's known that in brain development, abnormalities in size and shape can cause serious neurological disorders, and that include anencephaly, which involves missing part of the brain, hydrocephalus, which has the abnormal swelling of brain, etc. And so to look at the early brain development, we use a chick embryo as a model system. It's a vertebrate model but it's also it has significantly similar shapes to human development in the first 3 weeks. And so we use a fertilized egg, and we can culture it to a desirable stage. Is it still being recorded? Can you see it recorded?
Yeah, I think it's recording.
Okay. Yeah, I just couldn't see it. Okay, sorry. Yeah, so we can culture it in a desirable stage, and then you can see that initially the brain tube was a straight tube, and then it after 36 hours, and if we keep culturing it, it's subdivided into forebrain, midbrain, hindbrain, and at some point it starts to bend, and ventrally and also twist almost at the same time. So this progresses until the brain twists by almost 90 degrees. And if you now, let me show you this movie, how it works. So this is one of the earliest organ level left-right asymmetry events in development. So you can see that the heart is growing as well, and so the brain initially was left-right symmetric, but at some point the symmetry was broken, and it starts to twist rightwards, and until it forms almost a question mark. The question being, what are the forces that cause the brain to twist, and what determines the direction of twisting? Does bending affect torsion? So to address these questions, we start by making some observations. We use a technique called optical coherent tomography. So it uses laser to scan the cross section. So this is a cross section that you can see. So this is the brain tube. Above it has a membrane, it's called vitreous membrane, and underneath there's a heart, and then below the heart there's another membrane, it's called splanch nucleus, or SPL. So the brain tube is basically sandwiched between these two membranes, VM and SPL. And as the brain bends, it forms 3 fractures. There's a cranial fracture in the forebrain, and there's a cervical fracture in the hindbrain, and then there's a thoracic fracture that is in the spinal cord. And the thoracic fracture actually deforms it in the opposite direction. So we hypothesize that when the brain bends, it hits the vitreous membrane, and when it hits the vitreous membrane, it exerts a force that causes the brain to twist one way or the other. And if that is the only mechanism, then it should twist, 50-50 left and right. But why does it turns rightward majority? And we found that occasionally, so the heart normally loops to the right-hand side. Occasionally it forms a loop that goes to the left-hand side, and then the embryo also twists leftwards. And so that leads us to conjecture that perhaps there's a correlation between the heart looping direction and brain torsion. So these are our hypotheses, and talk about these first 2. And the third is that the degree of bending also affects the brain torsion. So to test these, we performed experiments in which we removed the vitreous membrane after about 40 hours of cultivation. And so you can see that the vitreous membrane now is no longer covering this part of the brain. And the torsion is also much reduced as compared to an embryo at a comparable stage. And now if we keep culturing the embryo for 11 hours, the brain twists a little bit more. So twist to about 43 degrees, but still is almost half of the twisting it otherwise would have achieved. And at this point, we can still apply an alternative force that can cause the brain to immediately twist to 90 degrees. And I'm going to show you on the next slide. And that shows the vitreous membrane does play a mechanical role in brain torsion. So in this video, I'm showing you, so we basically gradually lower the fluid level. So the brain is immersed in culture fluid. When we lower the fluid level, when the brain hits the fluid air interface, the surface tension kicks in, and that causes the brain to immediately twist to 90 degrees. So this happens in just a few seconds. This is in real time, and that shows it's mechanics that drives the brain torsion. Now, for the second hypothesis, since we conjecture the heart plays a mechanical role, we did microsurgery experiments where we pushed the heart from the right-hand side. So the heart here is on the right-hand side, there's a ventral view, to the left-hand side. And we found that if we now culture the embryo, the brain would twist leftward instead, as compared to a control embryo, it twists rightwards. So that happens every single time if this change of heart position happens early enough. But if we also found there's a point of no return. So if the brain already twists significantly, then pushing the heart to the left-hand side does not alter the direction of twisting of the brain. And so we were puzzled at the beginning, but later we realized that, oh, this cannot play. But we later realized basically, in this case, the heart, when we culture the embryo, the heart beats and it would return to the right-hand side in 1 to 2 minutes. So the heart cannot be kept on the left-hand side. So the twisting direction cannot be reversed. And we also built a finite element model using a simplified geometry of the brain. So the spinal cord is fixed on this end, and then we use another curve to represent the heart that goes to the right-hand side, and a block on top of the brain that represents the vitelline membrane. And as blanched nucleus is another block underneath the heart. And so I can show you this quasi-static loading. So we gradually apply a load from the top that represents the force from the vitelline membrane or from the surface tension, and the brain will twist rightward. And we also studied, did some parametric study to see the effects of some geometric parameters that include so alpha is the radius of the cranial flexure and the thoracic flexure. And then beta is the aspect ratio of the cross section. So we found that when we plot the torsional angle as a function of normalized position, so starting from this end, torsional angle is 0. So in all cases, it gradually progresses to almost 90 degrees. It takes off almost linearly to begin with, and then it plateaus. But if we use the parameters that is taken from the experiments, then the agreement is the best between our simulation and experiments. And the third, to test the third hypothesis about the relationship between bending and twisting, we use an implanted eyelash, which is much stiffer than the embryonic brain tissue, to inhibit bending. So we found that by still doing, the brain torsion is also significantly suppressed as compared to the control embryo. But if the implanted eyelash did not go far enough, so in other words, if it's only affecting the cranial flexure, not the cervical and thoracic flexure, then the brain torsion still can happen to almost 90 degrees. Now we can only see one eye. And that shows that the thoracic flexure and cervical flexure are more important for brain torsion. So to quickly sum up, we basically identified the mechanical origins of brain torsion, and that's related to the vitelline membrane exerting force and the heart positions being biased. And then there's correlation between bending and torsion. And then we have also been very interested in using principles learned from nature to design some biomimetic or bioinspired devices. So in this case, we tried to design a swimming robot that can be remotely controlled. And so you probably have seen the work from Kevin Kidparker's group. They designed some biomimetic swimming robots. That's fantastic. But they use pretty complicated techniques like optogenetics. But here we try to use a kind of a mechanical way of designing and controlling the embryo. So we use 3D printing to print out this swimming robot that takes on the shape of a airplane, but it's a swimming robot. And so we coded these striated patterns are the cardiomyocyte cells that's coded on the surface of PDMS. And so you can imagine this being a 2-layer system where the cardiomyocytes will periodically contract and relax. And that will lead to the bending or undulation of the fin. And that shows you how it moves. So if that is the only mechanism, however, the robot could only move forward, right? But how do we make it turn left or right? Okay, so this is a finite element simulation of this, what we call the cellular engine. So cellular engine means, we have these cells that periodically contract and relax, and that leads to the undulation, the bending. Okay, so that means, the cellular engine is on. So how do we turn it off? Or how do we make it turn left or right? So to do that, we're adding another layer of mechanism. So underneath the wing here, we coated a layer of photosensitive hydrogel. So it's a porous structure, and there's a gradient. If you zoom in, look at the microstructure, there's a gradient in the pore size across the thickness direction. And so finite element simulation shows that because of that, when it's heated, or when, in this case, when it's when we shine a near infrared light on it, it only bends in one direction. And so this is the second mechanism. So whenever we shoot near infrared light on it, it forms a curvature in the transverse direction. And so in this case, we also did finite element simulation. So using exactly the same strain for the cellular motion, we found that when the system adopts a transverse curvature, the bending rigidity becomes significantly large, much larger than before. And even though the cells are still contracting, relaxing, it only deforms the structure very minimally. And that basically is the way we shut off the cellular engine. So we can make it stop by shooting a near infrared light on it. And then we can also, so this is a comparison between the engine on and engine off. You can see that when the engine's on, so the strain is 8%, and there's significant bending, whereas when the engine's off, the bending deformation is minimal. And we also use this swimming robot to perform targeted drug delivery. So you can imagine that if we shoot the light unevenly, then we can control the left and right motion. And this is, so we use it to carry anti-cancer drug. So this is what we call the drug bomb. And we can precisely control it to be dropped off at 2 sites. And then observe that within a small radius, the majority of the cancer cells were killed. Yeah. So just a quick summary, we designed and fabricated biohybrid robot using cardiomyocyte cells, and then we mechanically turn on and off cellular engine, but that's in response to a near infrared light. Nice thing is that it can also penetrate tissues. So that gives a proof of concept for this type of remotely controllable swimming robot. We did mechanical analysis of mechanism and also showed a potential application in targeted drug delivery.
I have a conceptual question here. What is the maybe I missed it. What is the advantage of using this kind of hybrid model, using cells as this engine? Are they actually more efficient, or what is the logic behind?
I think so it's trying to take advantage of the best of both worlds, I guess, right? So the structure can be 3D printed, so we can design any structure we wanted. That's the synthetic part, and then we also use the biological materials. So that is probably like, it works well in the human bodies or animal models.
So what is the environment? Because those cells are pretty sensitive, fragile, right? So you can't really put it into a harsh environment.
Yeah.
So then what would be the niche application, so to speak?
So I think that would be application related to biomedical, like in human bodies or animal bodies.
Okay, not for like underwater robotics, that type of thing.
So the one that I showed was not for working under extreme conditions, but I think the same mechanism could be used, could be borrowed to design robots that could work under extreme conditions. So the idea of tuning, bending rigidity. So if you use shape memory,
I see the conceptual
memory material or other.
I see the mechanical concept here, but just practically, if you're working with very sensitive, like heart beating cell, like what kind of environment you can actually use them, just.
Yeah, they have a pretty narrow range of temperature, right? It can be functioning, but I think human body or the body of animals would be perfect.
Okay, gotcha. Thank you.
Yeah. Okay, so last but not least, I will walk you through collective cell migration very quickly. I think Jeff Berger already talked about this a while ago. Actually, it was he gave the first seminar on this topic, so I probably won't have to introduce too much, except to mention that it's still fascinating to think about cells, whether cells they behave more like a liquid or fluid, right? Sorry, liquid or solid. And so you have seen all these movies, I'm pretty sure. So wound healing also involves collective cell migration, and in encapsulation, we also see massive scale collective cell migration. Is this working? In the imagination. So in cancer, we know that EMT or epithelial to mesenchymal transition is very important, but there have been some recent papers that start to challenge whether they are required for metastasis. So the question now is, if it is not EMT, could it be partial EMT or could it be some other mechanisms? And that is why physicists have been trying to apply the concept of unjamming transition and see if that could be a more universal mechanism underlying the cancer cell invasion. And so inner particles such as coffee beans, can be jammed, right, as the density increases, for example. And it has been shown that in cells, adhesion, density, and motility are important factors. And so Dr. Faber proposed this phase diagram for jamming transition or unjamming transition. So this is an example of jamming, right? So people being pushed into packed trains. And this is not an example of unjamming transition. Is MCF10A cells that unjams upon the addition of RAB5A. So there are a number of factors that can induce this. But we're interested in mechanics and geometry, right? So we look at the shape of the cells in this case. So B and Manning, they found that when cells undergo unjamming transition, the shape characteristics change. So they defined a parameter that is cell perimeter divided by cell area to be the shape index. And they found that 3.81 to be a magic number. So when it's under 3.81, it's jammed. When it goes beyond that, it becomes unjammed. And people also realize that when it gets unjammed, it tends to take on an elongated shape. And so they proposed this model that write down. So they write down the total energy as the contribution from the incompatibility, 3D incompatibility, or resistance to the height fluctuation. So this you can think of it as the resistance to shape change, right? There's a penalty for doing that, and there's also resistance to the change of the cell parameter, and you can also think of them as linear springs. And so alternatively, you can write it in a non-dimensionalized way as follows. And then, so the P naught here is the cell shape index that I talked about. And it turns out this is a military secret 3.81 because it's exactly what the Pentagon has, right? The cell in shape index is 3.81. So, and there's also the T1 transition that occurs in this case. So cell can exchange neighbors during their migration. So they use a vertex model to study this unjamming transition. I mean, it's all elegant and quite nice, and the prediction actually also have been validated using the asthma cells, epithelial cells model. But there are also some things that I'm not quite satisfied with. So for example, vertex model only works for confluent cell layers. And then the T1 transition does not occur naturally. There's some, mathematical rules or like artificial rules you have to set up. And then there's also the limitation of the boundary condition. They use periodic boundary condition, and more importantly, the cell membranes here are not quite physical. So in the vertex model, it's simply it's like drawing a line, right, between two students. So once you form the centers of the cells, you draw a line between them to reform the cell boundary or cell membrane. So they do not physically move along with the cells. But in reality, I think it should be more like this. So between two cells, there should be two boundaries, right? One belong to each of the cells, and they should move along with the cells when they deform and migrate. And so that's why I started out to try to develop more universal computational method for simulating collective cell migration, and that would hopefully also work for wound healing and morphogenesis. And then also try to identify the roles of biophysical factors in engineering transitions. So the model setup is we think of cells as deformable polygons, and each cell has its own membrane, and then they should not penetrate each other or self-penetrate. And then we just need to prescribe the appropriate mechanical forces and mechanical properties depending on the systems we investigate. So started out as a Vernoid tessellation, and then we shrink the cells a little bit to form the boundaries for each of them. And then so each boundary of the cells is discretized into several points, and that way the cells can cell membrane do not need to be straight necessarily. And then the next step is to prescribe forces for the model. So here we put in also the linear restoring forces. So think of each cell boundary as linear springs. So if it deviates from the rest length, then there's a restoring force, and there's a restoring force. And then for each cell, when it deviates from the equilibrium shape, there's also a penalty. So there should also be another restoring force that drives it towards the equilibrium shape. And then since the cells we think of the cells as a viscous fluid, as a viscous media, we also have a viscous force term. And then in order to prevent the cells from penetrating each other, we also prescribed a Lennard-Jones type of potential. So that force will prevent the cells from penetrating each other. And then I'm going to quickly show you some of the simulations. So we can see that under this computational framework, the T1 transition can naturally occur. So cells initially do not contact. They can start to form new contact as they move along. So we don't need special rules to dictate how they undergo T1 transition. Yeah, so and in collaboration with Jeff Freiberg, Mingo, and Muhammad Zaman, we also have been modifying our model to validate their experimental findings. So, for example, they found that in MDA and MB231 cells, when there's these cells were embedded in collagen gel, when you increase when you change the density, there's a change of the behavior. So MCF10A cells, for example, they're more solid-like, so there are two ways to change these collective cell migration modes. So one way is to change the density of the matrix, so stiffness of the matrix, and the other is to change the motility of the cells. So in one case, they will form these finger-like patterns. So that's reminiscent of melting, and that's why we call it a solid-liquid transition. And in another scenario, cells can come out individually. So that is reminiscent of gas molecules. So that's more like a solid gas transition. And we also, using our computational model, we also simulate these scenarios. So we use these green, impenetrable, immobile particles to represent the matrix stiffness. So the denser these are, the higher the stiffness is. And we can also tune the cell motility to observe these transitions, phase transitions. I don't have time to talk about the phase diagram, but yeah, you can check out the. I'm actually gonna check out our paper online. Actually, that was on archive. And just very briefly, I'm gonna also show you that using this model system, we can also simulate wound healing. So the healing of the wound using this hybrid model, or the gas relation in Drosophila. So we just modify our model towards this new application. So hopefully, this model can be a more universal method that can be used to simulate a variety of different systems.
See, can I have a more specific question about, for example, here, when you try to model gastrulation, this imagination process, how do you actually simulate this process?
So in this case, I think what we did is we provided some driving forces to represent the apical constriction. So when the apical constriction happens, all the other cells will passively deform, and that leads to this gradual change of shape that represents the gastrulation. So this is still work in progress. We've been working with Binky at Dartmouth.
So I'm trying to figure out the uniqueness of the simulation program. Is that because now you can subscribe the differential behavior on different part of the vortex cell that you can simulate the different behavior?
Well, in this case, we basically look at a few cells near where the apical constriction happens. Yeah. So we can tune the rest length of the cells, and that can make it imaginate. And so we show that a local change in cell shapes will lead to a global deformation. Yeah.
See.
Okay. Yeah, so
sorry, the related question.
Sure.
I just want to clarify. So you do have to tune the local properties. Does that include the mechanical properties, or it's really just the shape?
Yeah, these are two things. I think the first is we changed the rest length that basically leads to the imagination. And the second is the change of mechanical properties, which we're still working on, because we found some interesting phenomena in the mutants, and we suspect that the mechanical properties may be different. And so that's why we also want to change the mechanical properties and see whether we can replicate that. Yeah, but that's still work in progress.
Yep.
Okay. All right, yeah, so just quickly sum up, we've been studying mechanics of formation of chiral structures in, helical ribbons or in the brain torsion. And then we also designed bioinspired soft robots and then studied collected cell migration. Yeah, and I'd like to, this is another paper we published on kind of bioinspired robots, where we use a electrostatic actuator to drive this electrostatic robots that can go back and forth with a bistable leg. Yeah, this is our philosophy, theory, and practice. So theory is what you know when you know everything but nothing works. Practice is when everything works but nobody knows why. In our lab, theory and practice are combined. Nobody works and nobody knows why. And of course, this joke, we strive to understand how things work in nature, right? And then we as an engineer and innovator, we try to build functional structures that works even better. And with that, I'd like to thank my students, postdocs, and my wonderful collaborators. And some of them are here. So Jeff, Ming, and Steve. Yeah. Thank you very much for your attention, and I'd be happy to address any other questions.
I have maybe a physics question. So in your model for the collective cell migration, can the model be, I don't know, somehow cross-grained and being those renormalization group kind of treatment to see whether you will see some new attraction basin or something like that? Or has it been done? Are you aware? Any of those model? Because apparently your model and say Lisa Menning's model are quite different, right? But I think in terms of describing behaviors, both have quite a bit of success. So maybe some of the parameters, if you just renormalize it, they'll just go somewhere else or something. I don't know.
Yeah, I haven't thought about that, but I think their model, I tend to think of their model as more phenomenological, and ours is more mechanical.
Sure. I see that. I'm just saying maybe. Yeah. Maybe
so
somehow
there should be
maybe you somehow renormalize them and they'll kind of go to the same place or something. I don't know. I'm just
right. It's possible. Yeah. But yeah, I would welcome any thoughts from anyone. I haven't thought about that.
All right.
It's a great point. Any other questions?
Well, I can just make a comment about your last slide about theory and practice. There's an old quote that goes like this. It says, yes, it works in practice, but does it work in theory?
That's fun.
Yeah.
I thought theorists usually find a way to make whatever works in practice work in theory, but maybe I'm wrong.
Yeah.
Yeah. Yeah. Sorry, I have to rush through some of the parts, but I try to cover as many as I can.
I would just say, Z, I'm not sure if I'd agree that about the vertex model is phenomenological. There's a lot of pretty deep physics that come out of that.
Okay.
So it's a lot more than curve fitting. And the nice thing is that kind of it gets at the point that was just made, that in their model, it turns out it all boils down, well, not all, but a lot of it boils down to this P0 parameter, this effective perimeter, which that one parameter takes into account stiffness of the cytoskeleton, cytoskeletal tension, and adhesion, all boiled in one parameter that's the key parameter. And this critical shape factor, 3.81. So that's pretty remarkable.
Yeah, I was saying it's elegant. Yeah.
Okay, good.
Yeah, I was saying it, use very simple assumptions and then arrive at some amazing results. That's pretty remarkable.
Z, I had a quick question. In these models, do the different cell types, do you consider the different properties of the cell types, or is every cell considered the same?
So that's a good question. I mean, when we set it up, we didn't differentiate the cell types, but I mean, depending on the systems, right? If we have a system with, a mixture of two types of cells or something like that, we can certainly do that. I mean, there's no reason every cell has to be the same.
Okay.
It's just for the systems that we simulate so far, we kind of assume they're the same.
Yeah.
Great. But we have
actually there's two papers that kind of relate to that. One is actually Max B and a man named Lee and Das have a paper in Physical Review Letters, I think last year, where they put in heterogeneous properties of cells. And looked at rigidity percolation in that system. And it's very interesting because they found that when the system is heterogeneous, that the pattern of contact percolation is not the same as the pattern of tension percolation. So that's what you see when you have heterogeneous systems. So that has been looked at. And another paper just appeared a few weeks ago in Cell from Eduard Hanizo and Jean-Philippe, I'm blanking on his last name, I shouldn't, it's a famous name, Heisenberg, about looking at the blastoderm in the zebrafish. And they actually focused on a lot of free space when the system when the cells aren't filling all space. What happens with rigidity percolation? Really interesting paper.
Yeah, I should look into that. Yeah, thanks.
Yeah, good.
So yeah, thanks for your talk. You give a very nice talk. I learned a lot.
Thanks.
Yeah, and I saw your helix. That's very interesting since you define 3 parameters. Yeah, K1, K2, and also theta. And I just imagine for the protein, there's also very similar helix structures for the protein, and it's not fully understood for the structure for protein.
Yeah.
We have an idea for applying your theory and idea to the protein structure, like that.
Yeah, I thought about that. I actually was thinking it'd be great if we have a protein folding person to work with. But yeah, let me know if you think of someone. That would be great.
Okay. Yeah.
Yeah, I mean, for the institute, if we can have someone working in that space, I think it would be nice. Yeah. Are there any